Welcome to MOOC courses on Controlled Drug Delivery Systems, Lecture 4, which constitutes Unit 1 of Novel Drug Delivery System BP 704T. Myself, Dr. Chitra Gupta, Associate Professor, Department of Pharmacy, Lloyd Institute of Management and Technology, Greater Noida. So, I have been talking to you about controlled drug delivery systems, the various aspects of it including the concepts. Then we discussed the factors affecting the designing of such systems, and then we went on to the various drug properties which affect the selection of drug candidate for formulating controlled drug delivery systems. Next, we go on to the approaches used in design of such systems. So, the approaches used to design such systems or the mechanisms employed in formulating such systems are categorized as follows. As shown on the screen, there are several mechanisms. First one are the dissolution controlled systems, which may be either matrix types or encapsulated type. The second ones are the diffusion controlled systems, which are matrix or reservoir types. Then we have got dissolution and diffusion controlled release systems. Fourth one, the water permeation controlled systems. These are based on either swelling mechanism, where the polymer is such which swells in contact with biological fluids and hence regulates the release of the drug, or osmotically controlled systems, which will be based on osmosis. Then we have got methods used using ion exchange resin, where the drug is absorbed onto the surface of an inert resins. These are utilized in delivery of ionic drug substances. The drug absorbed onto the surface of this resin is delivered as a controlled drug delivery system into the body. Inside the body, the drug is exchanged for similar ions present in the body fluids. Then we have got chemically controlled release systems. Chemically controlled release systems basically are based on polymers which undergo chemical transformations in contact with biological fluids such that the release of the drug is controlled. Next, we have pH independent formulations. These are the formulations which will release the drug independent of the pH of the environment. These are best exemplified by the buffered tablets. Another type of controlled drug delivery systems are the hydrogel preparations. These hydrogels are basically such polymers which will form gel when in contact with body fluids, and these gels, due to extensive cross linkage, will slow down the drug release when inside the body. Lastly, we have altered density formulations. They attain controlled drug delivery by enhancing the residence time of the drug in the body. So, we will discuss some of these mechanisms in details. Today's focus will be on the first two mechanisms, that is dissolution controlled matrix or encapsulated type systems and diffusion controlled matrix or reservoir type of systems. So, let us look at the dissolution controlled systems. The word dissolution itself implies that these are the systems which are based on slowing down the dissolution rate of the drug. So, the first point that has been mentioned here is that these systems are particularly useful for drugs with high aqueous solubility and high dissolution rates, because otherwise these drugs will be readily available for absorption in the body. In a controlled drug delivery system, basically we want to slow down the release of the drug, slow down the availability of the drug for absorption purpose. So, dissolution process, if you recall, that dissolution involved two steps. Supposing if this is the dissolving drug particle, initially there will be a formation of a stationary diffusion layer around the dissolving drug particles such that this stationary diffusion layer is saturated with the drug, and then there will be diffusion of drug from this stationary diffusion layer into the bulk of the liquid, such that the bulk of the liquid has a concentration which is much lesser than the concentration of the drug in the stationary diffusion layer around the drug particles. So, this dissolution involves two steps, we can say: the formation of the stationary diffusion layer around the dissolving drug particles, and secondly the diffusion of the drug from stationary diffusion layer into the bulk of the liquid. The second step, this is the second step, that is the diffusion of drug from the stationary diffusion layer into the bulk of the liquid, is the rate controlling step or the slower step in the process. Having understood the dissolution process, let us look at how these dissolution controlled systems are devised. So, in dissolution controlled systems, the dissolution rate of otherwise highly aqueous soluble drug or drug with high dissolution rate is slowed down by incorporating the drug in an insoluble polymeric matrix, leading to matrix type of systems, or the second approach that can be adopted is by coating the drug particles or granules with a polymeric material which will be insoluble, and the thickness of the polymeric material and the slow dissolution rate of the polymeric material will then govern the release of the drug from the dosage form. The rate of dissolution at steady state is expressed with the help of Navier-Whitney equation. You can see this equation here, dC by dt, where dC by dt gives you the rate of drug dissolution. This is equal to Ka by V into Cs minus C, where K is the partition coefficient of the drug, A is the surface area of the dissolving drug, V in the denominator represents the volume of the dissolution fluid, and Cs is the concentration of the drug in the bulk stationary diffusion layer, and C is the concentration of the drug in bulk of the liquid. So, we can see here that the dissolution rate is directly proportional to the partition coefficient of the drug and the surface area of the dissolving drug, whereas it is also dependent upon the concentration gradient. The Cs minus C basically represents the concentration gradient, that is delta C between the stationary diffusion layer and the bulk of the liquid. You must have studied that during dissolution testing of the drug, the dissolution medium is kept in constant agitation. That is, one of the reasons for that is that the concentration gradient has to be maintained. There should be no concentration build up in the bulk of the fluid. Another way of representing the same equation is dC by dt is equal to capital dA by H into Cs minus C, where D again represents the diffusion coefficient of the drug, A is the surface area, H represents the thickness of the stationary diffusion layer. So, the rate of dissolution is inversely proportional to the thickness of the stationary diffusion layer. Again, the agitation serves that purpose only. It removes the stationary diffusion layer. The agitation in the liquid, this keeps the thickness of the stationary diffusion layer to minimum. Then Cs minus C is the concentration gradient again, and it exhibits direct proportionality to the concentration gradient. So, as I told you, there are two types of dissolution controlled systems, matrix type and reservoir type. The basic system, that is matrix type. Here the drug is homogeneously dispersed throughout a rate controlling membrane. The polymeric membrane is called as the rate controlling membrane here, because it will slow down the dissolution of otherwise highly soluble drug. Example of this is compressing the drug with a slowly dissolving carrier into a tablet. The simplest matrix type of system is a tablet, which can be made by compressing a highly water soluble drug with some waxy material like carnauba wax or castor oil or beeswax. So, we can get a system which is where the drug is uniformly dispersed in a polymeric matrix, such that this polymeric matrix is water repellent or hydrophobic in nature. The water repellent property of this matrix is important, because it will prevent the drug from coming in contact with the dissolution medium and hence dissolving. So, because of this water repellent matrix, the dissolution rate of the drug is slowed down. The drug release from such systems occurs by first order kinetics, and rate of dissolution may be further controlled by altering the porosity of the system or by decreasing its wettability. Again, if the porosity is decreased, the interaction between water and the dissolving drug particles can be minimized. So, it is attempted that by altering the porosity, you can control the dissolution rate of the drug. Secondly, by decreasing the wettability of the matrix, again you can decrease the dissolution of the drug and its contact with the dissolution medium. Going on to the next systems, that are the reservoir type of dissolution controlled systems. Here the drug particles are coated or encapsulated by micro encapsulation technique. So, it is something like this. This is a drug particle. It will be enclosed inside a polymeric material coat. So, this is the core, which is made up of the drug, and this is polymeric coat. Now, since this coating prevents the drug particle from coming in direct contact with dissolution medium, the dissolution of otherwise highly water soluble drug will be slowed down. This polymeric coat acts as a barrier between the drug particles and the dissolution medium. Now, the rate of release of drug from this system will depend upon the thickness of this coat. Larger the thickness of the coat, slower will be the dissolution. Secondly, it will depend upon the rate of dissolution of this polymeric coat. So, these two parameters become important when we are formulating reservoir type of dissolution controlled systems. These are best exemplified by coated dosage forms, where by varying the thickness of the coat or material of the coat, we can accomplish different dissolution rates. In case of capsules, simple conventional capsules also, the dissolution can be slowed down by formaldehyde treatment of hard gelatin or soft gelatin capsules. However, once this coat dissolves or the capsule shell dissolves, all the drug will be released at once. So, the drug release, as I said, is proportional to the dissolution rate of the coating material, and it is inversely proportional to the thickness of the coat. Examples of some slowly dissolving coating material are cellulose, polyethylene glycols, polymethylene acrylates and waxes. This is a diagrammatic representation of reservoir type of systems, dissolution control systems, and matrix types of control release systems. You can see here clearly that in the first diagram, which represents a reservoir type of a system, there is a central core made up of the drug particles, and there is a coating membrane which is made up of the polymer. And as stated earlier, the drug release will depend upon the thickness of the coat and dissolution rate of the membrane coat. So, in case of reservoir type of dissolution controlled system, the drug release depends upon two parameters. What are they? The thickness of the coating and the rate of dissolution of the coat. Next, we go on to the polymeric matrix type of systems, which are again dissolution controlled systems we are talking about. So, in case of matrix type of system, the drug is uniformly dispersed in a polymeric matrix. So, you can see here this is a polymeric matrix, and drug particles are uniformly dispersed in this polymeric matrix. Now, the rate of drug release in this case will depend upon the rate of erosion of the core, that is the rate at which this matrix will break down or the matrix will dissolve, that will be the rate controlling step. So, that is about the dissolution type of systems, controlled drug delivery systems, where the dissolution rate is the rate controlling step in drug release, and we have studied two dissolution type of systems, that is reservoir types of systems and matrix types of systems. Going on to the next category or next approach towards development of controlled drug delivery systems, these are the diffusion controlled systems. Now, diffusion as a mechanism basically refers to spontaneous movement of drug molecules from a region of high concentration to low concentration, and the rate of diffusion or the flux of the drugs is represented by the symbol J, and this J, this flux across a membrane, is given by Fick's first law of diffusion. According to Fick's first law of diffusion, the rate of flux depends upon the diffusion coefficient of the drug and the concentration gradient along the barrier membrane. So, here you can see flux is directly proportional to the diffusion coefficient D of the drug, and it is also directly related to the change in concentration with the distance x of the drug. Supposing we are talking of a reservoir type of a system, diffusion controlled reservoir type of a system, in that case this distance x will be the thickness of the coat. If we are talking about a matrix type of a system, as the drug is released from the outer layers of the matrix, the outer layers will become depleted and the distance move will increase, will progressively increase, right. So, the distance will increase with time in a matrix type of system also. So, this concentration change with distance is important in controlling the release from diffusion controlled systems. The release rate here depends upon the diffusion through the inert water insoluble membrane barriers. Again, these diffusion controlled systems are of two types. We have got reservoir type of systems and matrix type of systems. In reservoir types of systems, water insoluble polymer material encloses a core of drug which then controls the rate of release of the drug. Matrix type of system, in this case the solid drug is homogeneously dispersed in an insoluble matrix and rate of drug release is governed by rate of drug diffusion and not by drug dissolution. In earlier systems where we were talking about dissolution controlled matrix type of systems, there the rate of release was dependent upon the rate of erosion of the matrix, but here it is dependent upon the diffusion of the drug, the rate of diffusion of the drug along the polymeric matrix, across the polymeric matrix. So, in reservoir type of systems, as I earlier mentioned, a water insoluble polymeric material encloses a core of drug which controls the release rate. Now, what happens that the drug will first partition itself into the coating membrane, and this partitioning of drug into the coating membrane is important because it in turn depends upon the partition coefficient of the drug, and then the drug present in the membrane would be exchanged with the fluid surrounding the particles. This is followed by more of drug partitioning into the membrane and then diffusing outwards, right. Examples of polymers used for making reservoir type of diffusion control release systems are ethyl cellulose and polyvinyl acetate. The rate of drug release in such systems is mathematically represented by the equation dm by dt is equal to A dk into delta C by L, where A represents the surface area available for diffusion. Larger the surface area available for diffusion, greater is the rate of drug release. D represents the diffusion coefficient of the drug, and this diffusion coefficient is in turn dependent upon the molecular weight of the drug, the shape of the drug, the size of the drug, right. Next parameter is the partition coefficient of the drug. This partition coefficient basically represents the partitioning ability of the drug between the core and the membrane and the polymeric membrane. So, higher this partition coefficient value, faster will be the drug release. Then next is the parameter L, which is the path length for diffusion. Basically, this path length, longer the path length, slower will be the rate of diffusion. Delta C represents the concentration gradient across the membrane available for diffusion of drug. Advantages of reservoir type of systems. In reservoir type of systems, the main advantage is that they can be so designed that we can accomplish zero order drug delivery. Secondly, the release rate can be varied by varying the polymer layer thickness or the nature of the polymer. The main disadvantage of reservoir type of systems is that these systems have to be physically removed from the implant site, and they are difficult to deliver high molecular weight compounds as shown in the earlier slide, because the rate depends upon the diffusion rate depends upon the molecular weight of the drug. So, they will be difficult to formulate for high molecular weight compounds. Diffusion controlled matrix type of systems. In these systems, a solid drug is homogeneously dispersed in an insoluble polymeric matrix, and the rate of release is governed by rate of drug diffusion and not by rate of drug dissolution. the rate of release can be expressed in different ways. For example, if a drug is dissolved in the matrix, then the rate of release is given by fixed second law of diffusion. If the drug is insoluble and it is dispersed in the matrix, it can be expressed with the help of fixed first law. If the matrix happens to be porous, then it will exhibit Higuchi's release, and if the matrix is hydrophilic, then it will exhibit release by gelation and diffusion mechanisms. So, these 4 different types of release can be observed with diffusion controlled matrix drug delivery systems. These diffusion controlled matrix drug delivery systems can be of 2 types further. The matrix can be rigid, for example, the matrix made up of PVP and fatty acids, or the matrix can be soluble matrix or glassy hydrogels for sustaining the release of highly water soluble drugs. Advantages of these matrix type of drug delivery systems are that these matrix are easy to produce, they can deliver high molecular weight compound. This was a restriction in reservoir type of delivery systems. Then their disadvantages are also there, that they cannot provide zero order release. The release from matrix systems is mostly first order release, because the rate of release will be dependent upon the residual concentration of the drug in the matrix. And lastly, the removal of the remaining matrix is required. If the matrix is not biodegradable, then it needs to be removed from the body. You can look at the diagrammatic representations of this system. Here, the first diagram represents the reservoir type of a device, where a drug core is enclosed inside a polymeric membrane, and I said that the thickness of the membrane controls the rate of release of the drug. Now, here you can see in the second diagram, at time 0, this is the state, at time greater than 0 and at any later time, the drug is first diffusing from the core into the membrane, and then from the membrane it is diffusing into the bulk of the medium. Here you can see a matrix type of a device. The outer layer of matrix will be first depleted of the drug by drug release from the outer layer, and then the drug would be replenished into the outer layer of the matrix by means of diffusion from the matrix. So, this is how the control of drug release is accomplished. Lastly, let us have a comparison between matrix and reservoir types of systems. Matrix systems are easy to fabricate. They have no danger of dose dumping. This is an important part, because the drug is embedded in the matrix, whereas in case of reservoir systems, the rupture of the coating membrane can lead to dose dumping. Then they can matrix system can give first order release. Zero order release rate is difficult to attain with matrix type of systems, but with reservoir type of systems, achievement of zero order rate is easy. Then matrix systems can be suitable for both biodegradable and non-biodegradable systems, whereas reservoir systems with degradability are difficult to design. Then in matrix system, one of the major disadvantages is that sometimes the drug on blending with the matrix may get adhered to the polymeric material and may get inactivated. This drawback is not there in reservoir systems, that the drug remains safe and in active form, it is not inactivated by the polymer. So, with this we can say that these two mechanisms are extremely important for formulating controlled drug delivery systems. In my next class, I will take up a few more mechanisms for drug delivery. Thank you very much for your patient listening.